everyone. Thank you so much for joining us today for a SupplySide Fresh webinar on the future of clinical research, centralized and decentralized models. I'm Devin Golam. I'm editor at SupplySide Supplement Journal, and I'll be the moderator for today's webinar. We are recording this, so it will be available on demand for all SupplySide Fresh participants, whether you're in the food and beverage or the supplement course. And I am delighted to be joined by some outstanding experts for today's discussion. We have Suzanne Mitschke, CEO and co-founder of Citrus Labs. We have Xenia Framrose, CEO and founder of Alethios. And we have Dr. Doug Kallman, co-founder at Substantiation Sciences and an esteemed professor at Nova Southeastern University. So we are here today to talk about clinical trial design, specifically centralized and decentralized. So clinical trials have an important role in the functional food and supplement industries to demonstrate raw ingredients or finished products actually deliver on advertised benefits. They form the basis for claim substantiation, establishing efficacious dosages, and determining if benefits affect certain populations over others. Now, while decentralized trials are gaining in popularity, they're not perfect in all scenarios, and there's a lot of factors to keep in mind when it comes to clinical trial design. Now, we're not here to debate the merits of centralized versus decentralized, but rather to give you an educational overview of how both serve our needs and play a role in building a scientific portfolio for your ingredient or your finished product. In today's session, we hope to outline some key considerations, introduce proper terminology, and help you feel more confident discussing clinical trials as part of your role in the industry. So I have quite the list of questions I've prepared for our panel, but if you have any questions as things go on today, please type them into the Q&A box and we'll get to as many as we can, and we'll try to have a couple minutes at the end for any additional questions. So let's get started. First things first, let's define the gold standard randomized placebo-controlled trial. We often hear about RCTs being the gold standard. Suzanne, can you define what this is and why we think of it as the gold standard?
Of course. So what you just mentioned, right? An RCT is probably what you think of when you hear the term clinical trial. An RCT stands for randomized controlled trial, and it's a category with different study designs under it. And RCTs are seen as the most trustworthy way to test whether a product actually works or not. And the most common design of an RCT is what you might know as a double-blind placebo-controlled trial. And here you split people into two groups. One gets your product, the intervention. The other one is a sham version called a placebo, and nobody should know which group they are actually in. And in a double-blind approach, the research team also doesn't know. So the actual participant and the researcher doesn't know which group is which. And then you compare results. And this is also called a parallel arm design because you have two study arms running in parallel. And you can have 2 arms, 3 arms, even more than that, and you control the arm that doesn't take your product. So that is essentially the control group. And what that means is they also don't have to have a product. So you don't have to have a placebo. They can also, test against a competitor, current standard of care, nutrition guidelines, or again, no product at all. And the next thing is where it gets a little bit more complex. In an RCT, you can also have just one study arm. And this one study arm can be their own control. And this design is called a crossover study. And I think, Devin, we are talking about that in a second as well.
Yes, great. Thank you so much, Suzanne. Xenia, over to you. Can you break down the difference between centralized and decentralized trials?
Of course, or I can at least try. Can everyone, actually, I had just a glitch on my end. Can you guys see and hear me?
Yes, yes.
Wonderful. All right, so when you think of a centralized trial, think of often called a site-based trial as well. This is a physical location in which participants are coming to a brick and mortar location and all things happen at this one location. What that can often look like is a participant or a patient may come into a site for all of their blood work to be evaluated by a clinician, to fill out surveys, to potentially have imaging done. You can have what is known as a single site trial in which all of the activity happens at that one site, or you can have a multi-site trial where there are sites all around the country to ensure that you have a diverse representation in the population that you're studying. Most pharmaceutical trials, especially in phase 2 and phase 3, are multi-site large trials. Then you've got decentralized trials. Now, decentralized trials have been around for a long time. If you talk to anyone who's been in this space about 20 years, they will tell you, I've been doing decentralized trials forever. This is nothing new. And the truth is that elements of a decentralized trial have been around for a really long time. If you think about the experience of going into a physical site and doing your blood work, you may also have someone call you while you're not at that site and say, hey, how are you doing? Could you let me know how you're responding to certain symptoms? So elements of decentralization have been around for a very long time. What we've seen, and there are companies that have done that as well for the last 10 years, and certainly during the pandemic, the number of decentralized trials, both in pharmaceutical and non-pharmaceutical context, rose significantly. A decentralized trial means everything is happening within the comfort of a participant's home. So that means that they might be, I know, Devin, you had any trials today you want to share with us?
No.
um, but a decentralized trial means that you have infrastructure that allows a participant to participate from home. They do everything from surveys to connecting wearables to doing at-home biomarker tests, or maybe having a mobile phlebotomist come to their location to do blood work. All of that is what happens in a decentralized trial. I know Doug is going to talk about the benefits and limitations of both, but I'll also just say one bit is that in the middle there you have a hybrid trial, which is where some of the elements are decentralized and some of the elements are in person.
Perfect. Couldn't ask for a better lead into the next question, Doug. Can you tell us where a centralized trial would make more sense and a decentralized trial would make more sense? Under what circumstances warrants one versus the other? And as Xenia said, where would a hybrid be appropriate?
These are great questions. And having a 20 plus year history in clinical research, phase 1, first in human through phase 4 on pharmaceutical, and with foods, dietary supplements, beverages, and other regulated items. Let's take this in the context of healthy people, not a pharmaceutical type study.
Sure.
So, one example where a central location type of study makes more sense with, let's say, a sports nutrition study. So where we're looking at an intervention of whatever it might be, could be a dietary supplement, could be a food, but you're looking at something to do with exercise performance, that you're measuring peak power, that you're measuring VO2 max, a measure of cardiorespiratory fitness. You're measuring aspects of strength, or you're measuring that combined with reaction time and a few other things. That data you can't do on a Whoop, a wearable. Um, that kind of is in person. And now let's say that you're tying it to some potential mechanism of action. So you have people undergoing physical tests and blood draws, right? Those in that kind of capacity, a central location as a sport nutrition example uh, is worthwhile. Another example would be um, that can fall into both camps would be joint discomfort. So some studies really want to use objective tests, right, where you are going to be doing objective gold standard tests of range of motion, of something called the 6 minute walk test, get up and go, and a variety of other things, um, maybe along with imaging. Whereas you can also do that kind of joint discomfort looking at validated questionnaires, right? So depending upon what you're looking for, it could be a site, it really, or decentralized or a hybrid. And hybrids are beautiful. In my pharmaceutical research experience, it's been a lot hybrid. And even so with hybrid, we had some visits where subjects, participants in the study were coming to the offices for, it could have been neurological testing and blood draws, or some nutritional exercise testing. And then in the between time between visits, there was e-diaries that were used, kind of like handheld devices before the iPhones blew up and Androids blew up, these ePro diaries that were used. And, so you could capture real time things now that are much easier. So centralized, really, what I want to bring it down to is it depends upon the objectives of the study, um, as well as what are the tools that are being used to help determine, whether there is to determine the outcomes and objectives. What are the measures and tools? Both have advantages, and combined, they're great as well. I think sometimes we have to also discuss um, purpose. What was the
Absolutely.
purpose of the study?
Absolutely.
And I'm sorry, within that, within studies, decentralized and centralized, often early studies are proof of concept, smaller, so
Hmm.
that you could see, is your theory having any legs from a statistical standpoint, trends or other or significance or otherwise, or effect size, some type of measuring that says, yes, I should double down and let's go to a randomized, double-blind, placebo-controlled trial. So those you might start in an office with an academic group or an independent site or what have you, and then move to something that could be done hybrid or decentralized.
Sure. And then just a quick example of a decentralized trial where that would make sense. And you guys correct me if I'm wrong, sleep, because it's so much easier to track how someone sleeps in the comfort of their own home versus going to some crazy sterile hospital room. Plus, with the advent of wearables, which we'll talk about later, we can actually get really good sleep data in the comfort of our own home. So let's move on to other study designs that aren't RCT. And I know you talked a little bit about this already, Suzanne, but we'll give you an opportunity to elaborate on that. Where would a different model be more useful? Could you touch on perception studies, maybe? And are there certain trial models that are best served with food products as opposed to dietary supplements?
Yeah, also what Doug mentioned, right? An RCT isn't always the right first step. You can think of it like buying a house. You typically don't start with a mansion, right? There's something called a starter home for a reason. And there are smarter ways to also build your evidence as you grow. And that's great because then instead of one study, you can actually have, an entire range of studies and can tell people, hey, we tested this not only once, but several times. And the beauty with it is that you can also test different things with different study designs as well. And what you mentioned, right, there's something called consumer perception studies. You give people your product, ask structured questions, and get results. Like 9 out of 10 users said they slept better. It's real research. It's scientifically designed, but faster, more affordable than a full RCT. And it gives you language you can actually use in marketing. And that is like super important. Then there's a thing called a single group clinical trial. It's a real IRB approved clinical study. You can also publish a single group clinical trial in a journal. You can include biomarkers, wearable devices, whatever you want. This is, robust, especially if you involve biomarkers. What Doug mentioned with objective markers, objective data, and it's hard to argue that blood work changes, for example, or microbiome changes come from a placebo effect, for example. And then what I mentioned earlier a little bit is crossover studies that are part of the RCT family. And instead of needing two big groups, everyone, tries both the real product and the placebo or control group or an entirely different product at different times. It's the same person, both products. You need fewer people, which saves money. It does take a little bit longer, but for food products specifically, your claims are usually about experience, taste, texture, how it makes someone feel. You can involve blood work. You don't have to involve blood work. But perception study or a single group clinical trial is often the perfect fit for food products. And a young brand might get way more value out of a study, that's like 5K, 10K perception study, then fuels their retail pitch, Amazon listing, versus doing like a full scale RCT right at the beginning.
Perfect. Thank you so much. So we've talked a little bit about the design of the clinical trial, but now we've got to get people in it, right? So let's talk about trial populations. Xenia, what are the biggest things that people should keep in mind when they are enrolling and selecting participants for their studies?
I would say the first thing to keep in mind always is going to be how you are powering your study and doing a sample size collection. So volume in terms of how many people do we need enrolled in this trial, adherent in a certain way, completing the study without adverse events for us to be making any type of robust claims from this data. So, we can certainly go into the details on a sample size calculation, but before that, it's thinking about what is the actual endpoint of this study and what is the primary endpoint of the study, which determines that power calculation. Other than that, certainly, I think with food and nutraceuticals, you want a population that is representative of the population that you are either trying to market to or make claims about. That's one side of it. At the same time, if you're going to be making claims that are generalizable to the larger population, you want to have very strong representation of what the generalizable population looks like for your product. Those are two of the things that I think are incredibly important. When you look at a study design, and you're thinking about that, that's actually one of the interesting things between site-based trials and centralized trials and decentralized trials. Where is the population that you're looking at? Are you capturing someone in a real world lived context, which is much easier to do in a decentralized trial? Or do you require a significant amount of control over their environment for what you are testing? Very appropriate for a centralized trial. In general, I think one of the frameworks that we use when we're trying to think of design and population is we go back to sort of a very rudimentary way of looking at the scientific method, which is let's say we get results from a study, good or bad, doesn't matter, but significant results from a study. To what degree are those results likely to be the result of bias in the design or bias in the population selection? Or to what degree are these likely to be just the random result of chance? Those two actually map on to how we think about statistical analysis, right? Type 1 and type 2 error. But I think for me, as someone who looks at this every day and talks to people who are doing their very first study and doing their 500th, that's a good way to think about it. Am I choosing a population that is going to add bias to my study? Am I choosing a population that is not large enough or representative enough that this result could just be a random fluke? That's a good way to think about it.
I love that. Doug, can you tell us a little bit more about inclusion versus exclusion criteria and why it's so important that we clearly define both of those when we're making a trial?
Yes, thank you. Well, the inclusion and exclusion criteria from a larger vantage describes and details just whom we're going to include in a study and who we are not going to include or exclude from the study. And it provides a rationale and reasons when you look through the inclusion and exclusion criteria. So let me give an example. Let's say that we were doing a study and we wanted to do adults. Well, typically, we're going to define that. And do we want young adults? Do we want old adults? What are we defining as old, and will that offend somebody? Or older adults? And do we want both genders? And these type of things come into play. So the inclusion criteria help tell us, for example, we want people aged 21 to 55, both genders. And then there are other things that help clean up the criteria. With dietary supplements, amongst the major things to think about with inclusion and exclusion criteria is that it represents a healthy population, because dietary supplements are intended, right, for market to healthy, not to treat, prevent, cure, and the rest of that FDA disclaimer. So the inclusion criteria describes exactly whom we're going to include, who we're going to exclude, and definitions thereof. So for example, again, even in a healthy subject study, Let's say that you're doing some blood tests of normal metabolic panel and complete blood count for screening purposes, and you find an abnormal reading, something that requires a follow-up test. Well, that person might be screened out because you just, in my past, we've found cancer and other things in people just on screening blood tests that have had to been referred out. So those things, that's what the inclusion and exclusion criteria even help to make sure that it's a healthy population and that it's meeting whatever the objective of the study is.
That's a great lead into the next question that I have, Doug. One thing we do have to keep in mind is the outcomes that we're looking at because they are meant for healthy populations. So we have to make sure we're not testing things on diseased participants. Suzanne, can you tell us about some common misconceptions? Like what's a need state, like need states? Which ones are diseases? Which ones are more transient conditions? And what's the regulatory implications if we're testing on a diseased population?
Yeah, that's a very good topic. And when you're testing around like stress, bad sleep, low energy, bloating, these aren't diseases. They are normal temporary things, that healthy people experience. And that's exactly what supplements are designed to do, right? Keeping healthy people healthy. And the problem starts when you test your product, what Doug already mentioned, on people who have actual diagnosed medical conditions like clinical insomnia or even IBS. And now you enter territory. And the FDA does not really like it when supplements make drug level claims or even just research, their supplements on populations that actually have diseases. This could trigger something that is called an investigational new drug application, an IND, and you definitely don't want that. And a simple way to think about it, you should go after, like, I sometimes have trouble falling asleep, right? This is supplement territory versus my doctor diagnosed me with insomnia. This is drug territory. And the words you use in your study also to describe your participants, or even, what you are measuring, that matters a lot. And if you run a study on sick people and then use those results to market a supplement, you're basically building, a drug claim on a supplement label. Even if you're not making any claims, your study is, on that population, then this is a big issue. And, you just, raise your chances of getting a warning letter from the FDA or even the FTC.
This is why we have the experts here today, because we want to make sure when you're designing a clinical trial, obviously you're running that trial because you want to use the data to market your products. We need to make sure we get clean data to market effectively. And because we're talking about testing real live humans here, we'd be remiss if we didn't address ethics. So this is where an IRB comes in. Xenia, can you tell me what IRB stands for? I know, tell our audience though. And how does one go about getting approval for a study?
Wonderful. So an IRB is an institutional review board, which is a committee that has a number of individuals who are qualified together as a group to decide whether or not the science that you're trying to conduct is ethical and safe. That is primarily the responsibility of an IRB, safety of the participants. That's what it's there to do. There are different IRBs around the world and around the country. In Europe, known as an ethics committee. In the US, an IRB. There are academic IRBs. So Doug is probably very familiar with many academic IRBs, where it is university officials who sit on that committee and often are dealing with studies that are funded by grants. In industry, and that's pharma and non-pharma, most folks will use sort of central IRBs, which are sometimes not-for-profit, but sometimes function as for-profit organizations as well. So I think it's important to think about like the commercial underpinnings of these organizations as well, because it does determine how quickly an organization can move, what their liability is, and I think understanding that is important. I'm going to name some of the IRBs because this is an audience that I know is looking to learn, but often you will have on the pharmaceutical side, you've got Advara and WCG. In the dietary supplement space, I think we stay away from the WCGs and the Advaras. Suzanne, Doug, give me a nod if that's also your experience. Just because they're not as well equipped to deal with dietary supplement studies, or certainly not decentralized studies either. So often there are folks like Sterling, Brainy, Pearl, lots of central IRBs that you can use. So Devin, for the second half of your question, which is how do you go about getting IRB approval? An IRB needs to see all of the ways in which you are interacting with a participant in your study. So if that is a site-based study, they have to see, all right, this is the intake form that someone is going to use when the participant comes to the clinic. These are the documents we're going to use. If it's a decentralized study, and it's done on an app, they want to see screenshots of that. How are you going to be asking those questions? All those things are the manifestations of what is the most important document to submit to an IRB, which is your clinical protocol. A protocol contains all of the information, and there are certainly great templates out there all over the place. And there's a great one on the NIH website as well. But a protocol details what you're going to study, why you're going to study it, the rationale for the endpoints that you choose, the inclusion and exclusion criteria, how you're going to analyze the data in a statistical analysis plan, all the references that you have to make some of the claims for how you're going to go about this study. Other things that you submit along with the protocol are an informed consent form, a critically important document for an IRB submission, which details exactly how a participant's data is going to be used, what they're consenting to, crucially outlines the risks and benefits of potentially participating in this type of research. And then you've got a host of other things that will also go in there. Information on the product that is being studied. All of your recruitment materials for how you are planning to bring folks into your study. So an IRB will look at that, and let's think about sort of safety and ethical appropriateness, right? Recruitment materials are a great example of where all of that comes into play. You don't want to have something that says, join my joint health study. It's going to heal your knees and you're going to get $150. Probably not what you want to put out there. I know, Doug, I know. That's what all of mine say too. So,
Yeah.
you have to think about protecting this population. Like to be able to do research is an incredible privilege, and it is an incredible responsibility. And an IRB is usually alongside you to make sure that you're recognizing that that is very much the truth. Anything I missed there, Devin, that you want me to touch on?
I don't think so. The only thing I was going to add is that getting IRB approval is important regardless of the product that you're testing. So most people know I'm a food scientist, and when I was in grad school, I did a sensory test on some edible bean-based bars, and I had to get IRB approval just to feed that to people. Even though it's completely food grade, completely safe, I still had to prove to the IRB I wasn't going to kill anyone with my bean products. So,
Yeah.
so important regardless of what
I
you're doing.
just add there that if you're thinking like, do I need IRB approval for this study? If you're giving something to someone, chances are
Yes.
you need IRB approval. If you are giving something to someone in a highly controlled way, where you're like, take this on day 1 and day 7, that needs IRB approval. Things that
100.
don't need IRB approval. I want to study people's nutritional intake in an observational study over the course of 4 weeks. If you want to publish it, you definitely need IRB approval anyway, so that's important. But studies like that are observational sometimes will get an IRB exemption. There was one other thing I think I put in my notes that I wanted to touch. A lot of the studies in dietary supplements and functional foods will qualify for what would be two determinations: expedited review and a minimum risk. So because unless you're looking at a novel food or a new dietary ingredient that has really never been given to humans, or doesn't have a great risk profile, you will usually qualify for expedited review or minimum risk, and you have to justify those things to an IRB.
Great. All right, so we've got our study design. We have our people. Now we need to collect data, which is the stuff that I really like. So Before we get to this, I think it's important we understand the difference between subjective data versus objective data. And Doug, I'm going to tap you to tell us the difference between this and give us an example of each.
On a scale of 1 to 10, Devin, how do you feel where 1 equals blah and 10 equals unbelievable happiness with right at this moment?
11, because I'm with all of you.
Right. That is subjective data. That is
Yes.
an example of subjective data. Often in research, validated questionnaires are used rather to obtain subjective data. That these validated questionnaires most popularly used would fall under quality of life.
Yes.
So it could be anything ranging from mood states, positive and negative affect moods, aspects of moods, to stress levels, to perceived sleep quality, and so forth. So, subjective is really getting the person's response to something, even if it's in an ordinal format, like a Likert scale of 1 to 5 or visual analog scale, it's still subjective. Versus objective would be you go to your doctor's office, and some doctors will physically actually take the blood pressure themselves versus using an automated machine, which most use, and the bills are the same. But nonetheless, when the doctor is taking, even if it's an automated machine or he or she, as the physician, is taking the blood pressure, that is objective data, right? Because it's giving us a reading from something that is, in this instance, and for research, validated measures. So sometimes in nutrition studies, that could be the objective data could be through blood tests. It could be through cognitive testing. It could be through a variety of different types of tests where data is output. When you, Devin, were involved in sensory testing, that's subjective.
Yes.
And
Yes. 100%.
one of the things I just wanted to add to the IRB, since we're talking about research just back, is that um, we use IRBs historically for the safeguards of the public.
Yeah.
Right? And when in doubt, ask, do I need an IRB? The FDA does have a wonderful question and answer page on do I need an IRB? And am I, and or is this project potentially IRB exempt? So use the resources that are out there besides the people and research groups that are out there. Um,
Excellent.
so yeah, objectively, I think I've answered the question.
Yes, you have. So, speaking of objective data, Xenia, I know wearables are very much in your wheelhouse, and we're hearing
Wearables.
about them used so much more in study design. Can you tell us what wearables are being utilized most, and what sort of data are they providing scientists?
Yeah, I'm going to put wearables in a broader category that we internally call participant-generated health data. And
Okay.
that can really fall under, you know, wearables. And when I say wearables, I mean a Garmin, a Fitbit, an Oura Ring. Um, you know, I know we can't ask anyone, but I'm always interested in knowing what wearables people have because it's always it's a real indication of like how people do things. I know Suzanne is a huge athlete, and so I'm interested. I think you got an Aura ring, if I'm not mistaken, right?
It looked like she had something on her arm right
Oh,
there,
yes.
too. Yep. On her wrist.
So you've
Yep.
got commercially available wearables, you've got devices and sensors. So think you have a smart scale. If you have a continuous glucose monitor from a Dexcom or an Abbott Libre. And then I would add another third category, which is digital biomarkers. And people use that word in lots of different ways, but this is certainly a growing area where you can have AI models that could detect, do you have are you about to get COVID based on the sound of your voice? Are you experiencing a certain symptom of an illness based on the color of your skin? Lots of people spend a lot of time developing those digital biomarkers, and I think they're fascinating. So, what wearables are being used more often? This goes back to what Doug said at the beginning, which is what is the purpose of the study and are the endpoints appropriate for a decentralized study? Um, I would say almost all the studies that I've worked on have always collected sleep quality, even if just as an exploratory variable. It's one of the things that almost all of the devices will provide you with some degree of standardization. And I would say that the Apple Watch is the most popular device out there. And I'm going to say the worst device for sleep quality. So when you are choosing like what wearable you want to include and incorporate into a study, there are lots of ways in which you can um, bring that to the highest tier and quality of your research, or have it be something more exploratory where you're just looking for signal.
No.
What do I mean by that, right? If sleep's not your primary endpoint, you can have potentially a number of wearables in an observational study and see, okay, it looks like the best quality of data for my study is coming from Fitbits. You could also say, I want to make sure everyone has the same device for this menopause study, and I want to make sure I'm looking at body temperature in the right way. I'm going to give every single person an Oura ring. I'm going to add a period of time that lets me capture a baseline, what we would call a run-in period, so that I even know what the change from the baseline is in the study. So there are lots of ways to use wearables. It's very important to consider what the purpose of them is in your study. And if I had to say I have a favorite, I'm a Garmin girl.
Awesome. So, Suzanne, we know that questionnaires and wearables are great helping get data for decentralized trials, but there's some other measurements beyond wearables and questionnaires that help demonstrate health benefits. Can you give us an example of this type of data and where a centralized trial would make more sense over a decentralized?
So technically, any study can be entirely done from the comfort of the participant's home, especially standard supplements, food studies, some things people can do at home, surveys, right? Fitness tracker, blood draws, as we heard, right? Personally, I don't like these at-home finger prick or these stick-on blood tests because of user error. We've tested it extensively. Half of it, or even more than half of it, typically comes back as no analysis possible. And, obviously there are also things like microbiome, also very easily done at home, or body composition with smart scales, works great for a lot of studies. But some things are better in a lab. So it's a personal preference. A blood work, some people might not want a stranger in their house for home phlebotomy. They rather go somewhere. Better body composition with DEXA, for example, you again have to go somewhere. Proper before and after photos, specific skin measurements, hair measurements, anthropometrics, for example, like waist circumference measurements, or even when a study design is more complex. So, for example, we've done a bunch of bloating studies where people had to eat 2 slices of pizza. Obviously, that has to be controlled. It has to be the same pizza. It has to be exactly the same, amount slice. And then, measure bloating via waist circumference. This is actually very complex to measure waist circumference. So this has to be done by a specialist. And then also, we talked a little bit about me being an athlete, our specific performance metrics. But particularly in the sports performance field, you have to have a treadmill, a bike, right? It has to be supervised. Some specific lactate measurement has to be done, or strength, has to be done. So you need proper equipment and properly trained individuals to actually do that. But I like the best of both worlds that Doug already touched on. Hybrid trials, I think, is the way to go versus fully decentralized or fully centralized. There is as always, a good compromise, a good middle ground, because I personally don't believe in fully centralized studies anymore, simply because it is more participant burden. And for, we're talking to early stage companies right now, right? So it's centralized trials. Typically, you have to have like a significantly bigger budget compared to decentralized or hybrid.
Excellent. Great insights. So now that we've talked about collecting our data, we have to analyze it, right? That's where statistics comes in, which was probably my least favorite subject in grad school, I will freely admit. So, one thing that I see so much that people get hung up on is significant difference versus clinical relevance. Sometimes something will show a significant difference, but not necessarily mean much in the real world. So, Doug, I'm wondering if you can elaborate on this for us a little bit more and talk about the differences between statistical significance and clinical relevance.
Sure. So, statistical significance in research is typically defined as a p-value less than 0.05, which loosely we could think of 1 times out of 20 this occurs, because that
Yep.
equals 5%. And so automatically that means hitting that bar is going to be pretty high. So depending upon what the outcome is of interest, statistical significance may not have relevance. And in honor of Suzanne and her athletic heritage, um, I'll use sports as an example. There are a number of studies that have been done that will show various dietary supplements can enhance performance by about 1 to 3%, right? And you might not say, oh, that's not much. 1 to 3% might be not statistically significant in some of the studies. However, that actually translates for you have not medaled to now you're the gold medalist, because that 1 to 3% was actually fractions of a second that you were able to cut off the 100 meters.
Mm.
So that's one way of looking at. That's when in statistics, instead of doing statistics, or in addition to doing traditional statistics, you add in something that's called effect size analysis. And that gives you a better interpretation. Is this a meaningful change? Now, meaningful change also in research can apply to other things. And I know I previously mentioned quality of life uh, uh, scales and questionnaires that are used. So, for example, in the real world, um, for let's say the Hamilton Depression Scale is being used to score somebody's depression level. Well, in clinical practice, when somebody has a 50% reduction in that score, it's considered clinically meaningful. So, understanding what the change value of any of these questionnaires are help for understanding for is something meaningful, who cares about the p value. So, for example, if people that on a stress test, not stress physical, but on a stress questionnaire, if their score was reduced by 50%, that is something when we say meaningful, clinically meaningful, you would notice in your everyday life. Your therapist would notice in your everyday life. Your friends would. Where sometimes statistics aren't the whole story, but we do need to use traditional statistical analyses. We have to understand what is the right approach for the question being asked and the populations being analyzed and the outcomes being measured. So earlier I started that one out of a p-value of less than 0.05 generally means 1 out of every 20 times you're going to have that positive finding. So now, to add a little bit of conflict, the more questions you ask, sometimes you get false positives. So
Mm.
in research statistics, that you could control for that. So the best of both worlds is having a good understanding of what meaningful change is, what clinically meaningful change is, and how to apply the right traditional statistics with even effect size calculations, which are more real world relevant for a great variety of dietary supplement type studies.
I love that. I love the kind of the reverse example of how something might not be statistically significant, but still clinically relevant, because I always think about it in the reverse. And like one example is weight loss products. Like so many times I will read a clinical trial for a product and it will say that without any other changes in diet or exercise, someone has lost a kilo after 8 weeks, let's say. Our, like as a woman, like our weight fluctuates that much just going through our menstrual cycle. So is that really clinically relevant? No, even though the stats might say it's statistically significant, but I love that the reverse can also be true. I love it when I learn things on these webinars. That's fantastic, Doug. Thank you. So I know we're coming up a little bit on time, so I'm going to gloss over a couple of questions here. But I really want to hear from all of you. With all the things we discussed, what are the biggest mistakes you've seen carried out in clinical trials that anyone listening to this should avoid? So, Doug, let's go to you first, and then Suzanne and Xenia.
I don't know if it was biggest, but this was definitely an oops. Um, when you're going to sponsor and work on a clinical trial, and it's, let's say you are the company that is sponsoring it, make sure that all of the testing that is done on the product ahead of time and labeling is correct. In this instance, we did a study, and after the study, we did all the statistical analyses, and but as part of our SOPs, the retained product we sent out for analysis. And lo and behold, the manufacturer who put the labeling together on the product and the placebo for the project switched them.
Oh,
They did not follow they made a mistake somewhere. So everybody that was in the placebo group actually got,
jeez.
real product. Their labeling was wrong. That's a major mistake.
Yes,
That took a long time to unwind, but be able to unwind where everything was traceable to, like, that's what it was. We're not just switching anything.
crazy.
So that is a big oops. Quality control of product is very, very important, and labeling. Have a systems of checks and balances somehow there in there.
Absolutely. Suzanne, what about you?
So I think over-engineering a study is a big mistake that I'm seeing all the time, making study too complex unnecessarily just to sound more sophisticated, for example, or testing too many things at once, kind of what I call the spray and pray approach, to get like the most bang for your buck. And brands try to save money, testing on fewer people, having more endpoints. And that is typically where things go wrong, because then typically results are really wishy-washy.
That's so interesting. Thank you. Xenia, what about you?
Yeah, this is a corollary to what Suzanne just said, which is choosing the right sort of rung of, if like research is a ladder, and Devin, I know I say that all the time, but choosing to start on the right rung. You may not be ready for a massive double-blind placebo-controlled trial. There may not have been enough work done on the intervention to justify doing that. And then, you start there. You sort of pack that study with all types of like different forms of bias because you really want to get a really specific result, but you've never done the study before that gives you signal on all of the other possible results you might have been interested in or looking at. So I think choosing the right study design, there is no one answer for one product at any point in time, right? Like you've got it's got to be a match, and I think that's important.
Excellent. And then the last question I have for you guys, and we'll close us out with this. If someone came to you doing a new clinical trial for the first time, what is the one piece of advice you would want to give them? Doug.
Oh, never to put the spotlight on Doug first.
Too late.
Okay. So if somebody and they had no experience, no knowledge, no awareness, what would be the first thing that I would want to walk them, I would want to walk them through the process and help them manage expectations.
Hmm.
One of the biggest things that I see as occurring is, and I think Suzanne and Xenia mentioned it, people have these ideas, so they want to throw everything against the wall, but they don't realize at all what things cost, and that the accuracy and precision and reproducibility of various kinds of tests matter to give you veracity to your data. So I think really taking a measured approach and get as much knowledge leaders as you could find to help you um, put together your first study, which could be a proof of concept study, could be a preclinical study
Yeah.
that's done in a laboratory or done in another preclinical fashion, could be a small human study. In the pharmaceutical world, proof of concept studies are done on 8 to 12 people just to see if the drug that they think that may have an effect for something may have an effect. So there's no reason why in this world a small proof of concept study doesn't have validity too. Hope that helps.
Excellent. Suzanne, what's your top advice?
My top advice would be to start at the end. Start with what the goal of the study actually is. Is it marketing claims? Is it just to get an understanding how the product works? Is it that maybe your product's been on the market for a while and you have a couple of hypotheses that you want to test? So start at the end and think really about what you want to get out of the study, and then find a research partner to actually help deliver on your end product, your goal.
Excellent. Xenia, take us home.
It is almost the same and also the opposite of what Doug said. So I'm worried now. I know it's the first time we're disagreeing. So I think when you're a total novice, yes, it's so important to talk to experts and get a lot of opinions. But I talk to people who are total novices and seasoned researchers. And the best studies always come out when the person on the other side of the table for me is deeply inquisitive, right? Especially on things that, we do this all the time. We come from a place of authority and we're like, should be this measure, should be that. Be inquisitive with who you're working with and say, why would that be the case? I think in this space and in certainly in all research, jargon and opacity is a form of like keeping people out of the process of being partners in research. So do what you can to ask those questions. And certainly if you have Doug, myself, or Suzanne on the call, we'll be thrilled. We'll do a whole other session like this. But I think that is important because the hope is that it's not your only study when it's your first study. The hope is that you start somewhere and keep moving. And to do that, I think you have to be a true partner and be very inquisitive. So that's what I would recommend.
Excellent. Spoken like a true scientist asking questions. So, Suzanne, Doug, Xenia, thank you so much for joining me for this. What a great discussion. You have so much expertise, and I really appreciate you taking a little time today to share that with us. And as a reminder, this webinar will be on demand. So if you want to watch again and strengthen your understanding of clinical design, it'll be available in a week or so. On behalf of everyone here and myself, I'm Devin Golam of SupplySide Supplement Journal and SupplySide Fresh today. Thank you for your time. Have a great day and go make the world better one product at a time.